Good morning, and thank you all for attending today's Journal Club webinar sponsored by the ATS RSF Assembly. My name is Niyati Borkar from the Mayo Clinic, and I'll be moderating today's webinar titled When Inflammation Stresses the Airways, ER Stress Drives Airway Smooth Muscle Proliferation. It is my pleasure to introduce Dr. Debanjali Dasgupta, who is a postdoctoral research associate working under the mentorship of Dr. Gary Seek, here with us today. Dr. Dasgupta will be presenting her recent AJP lung publication on tumor necrosis factor or TNF alpha induced endoplasmic reticulum stress promotes airway smooth muscle cell proliferation. Dr. Dasgupta's work investigates how inflammatory mediators, particularly TNF alpha, trigger endoplasmic reticulum stress and activate proliferative signaling pathways in human airway smooth muscle cells. Please join me in warmly welcoming Dr. Debanjali Dasgupta. Joining me in co moderating today's session is Dr. Sergey Gutor from the University of Michigan. With that, I'll turn it over to Sergey, who will introduce our distinguished panelists.
It is my pleasure to introduce Dr. Sathish Venkatachalem. Dr. Sathish is a professor of medicine and a laureate chair in molecular medicine at the University of Oklahoma Health Campus, where he is also served as a co-director of research in the Division of Pulmonary Critical Care and Sleep Medicine. His research program focuses on diseases including asthma, COPD, pulmonary hypertension, and lymphangioliomyomatosis. In particular, his laboratory investigates how sex hormones, inflammatory signaling, and airway structural cells contribute to lung disease development and progression. His work combines fundamental mechanistic research with a strong transitional goal of identifying new biomarkers and therapeutic strategies for patients with chronic lung diseases. Dr. Sathish is also a dedicated and highly respected mentor. He has trained numerous graduate students, postdoctoral fellows, and early career scientists such as Niyati. And Dr. Satish's laboratory is very successful and supported by multiple NIH grants. So please join me in warmly welcoming Dr. Satish. Next, it is my pleasure to introduce Dr. Rodney Britt, an accomplished pulmonary scientist whose research is advancing our understanding how inflammation affects airway structure and function in asthma, cystic fibrosis, and bronchopulmonary dysplasia. Dr. Britt is an associate professor of pediatrics at the Ohio State University College of Medicine and a principal investigator in the Center for Perinatal Research at National Wild Children's Hospital. Dr. Britt's research focuses on the immunological and physiological mechanisms underlying chronic airway diseases, including severe asthma. His laboratory is particularly interested in understanding how inflammatory signaling alters airway structure and function and contributes to corticosteroid insensitivity. Using primary human airway cells, preclinical models, and molecular genomic approaches, his team seeks to identify novel strategies to improve corticosteroid responsiveness and reduce airway inflammation remodeling. In addition to his scientific contributions, Dr. Britt is strongly committed to mentoring and developing the next generation of biomedical researchers. He is also an American Thoracic Society Fellow, reflecting his contribution to pulmonary research and respiratory scientific community. So please join in me in warmly welcoming Dr. Britt.
Thank you, Sergey, and thank you, Dr. Satish and Dr. Britt, for joining us today. As a note, this session will be recorded and posted on the ATS website, and a recording link will also be sent to all the registrants. We will have approximately 30 minutes after Dr. Dasgupta's presentation for questions regarding the research, as well as questions pertaining to the overall topic for discussion with the experts. Please feel free to use the Zoom Q&A function to enter your questions at any time. With that, I will let Debanjali begin her presentation. Debanjali, the floor is yours.
Thank you, Niyati, for the nice introduction. Good morning, everyone. First, I would like to convey my thanks to Dr. Borkar for inviting me to present my paper here. And I would also want to convey my sincere gratitude to Dr. Britt and Dr. Sathish for joining the presentation as expert panelists. Today, I will be presenting the paper that we published maybe last year, December or early this year, that is entitled as Tumor Necrosis Factor α-Induced Endoplasmic Reticulum Stress Promotes Airway Smooth Muscle Cell Proliferation. This study mainly investigates how inflammatory signaling can promote airway smooth muscle proliferation through activation of endoplasmic reticulum stress pathway, specifically the IRE1 alpha mediated pathway. As the background of the study, airway remodeling includes structural changes in airway, such as smooth muscle hypertrophy, airway smooth muscle hyperplasia, and thickening of the airway wall. Among these changes, airway smooth muscle proliferation contributes directly to airway hyperplasia and narrowing of the airway that is related to airway hyperresponsiveness. TNF-alpha is one of the major pleiotropic pro-inflammatory cytokine that is reported to be elevated in airway inflammatory diseases, mostly in asthma, COPD, etc. And this TNF-alpha, we previously published multiple studies showing that TNF-alpha selectively activates IRE1 alpha phosphorylation and XPP1 splicing mediated endoplasmic reticulum stress pathway in human airway smooth muscle cells without any significant activation of two other endoplasmic reticulum stress pathways, which are activated by PARC and ATF6. I will come to all of these individual sensor proteins in my result section. This is just a brief description of how it works. And we also published that once TNF-alpha activates IRE1 alpha XPP1 excess of ear stress, XPP1 splice variant works as a transcription factor. And it transcriptionally activates several genes related to protein folding, just to diminish the effect of ear stress, ear homeostasis proteins, metabolism-related genes. We recently published XPP1 splice variant transcriptionally activates expression of cyclin-dependent kinases, CDK1 and CDK5. This CDK1 and CDK5, as its kinases, it can phosphorylate several other proteins. One of the major phosphorylating target is dynamin-related protein 1, and we also published that this CDK1 and CDK5 mediates phosphorylation of dynamin-related protein 1 and promotes mitochondrial fragmentation. Beside it, the role of CDK1 and CDK5 modulating the mitochondrial fragmentation, CDK1 and CDK5 are two major proteins plays role in cell cycle regulation. Cyclin B1 is a major cell cycle regulatory protein that forms complexes with CDK1 as well as CDK5. Cyclin B1 forms complex with CDK1 that is called MPF, mitosis promoting factor, and promotes G to M transition during cell cycle. Whereas CDK1 is the canonical molecule for cyclin B1 activation, whereas cyclin B1 also can form complex with CDK5. That complex mediates high fidelity error-free mitosis by ensuring proper segregation of chromosome during cell division. So based on this previous findings, we hypothesized for this study, TNF-alpha selectively activates IRE1 alpha and XPP1 splicing mediated ear stress signaling pathway, which transcriptionally might activate the expression of cyclin B1 along with CDK1 and CDK5. Then cyclin B1 can form complexes with CDK1 and CDK5 to promote proliferation of human ASM cells.
View this full video at atsnow.thoracic.org.